DNA is the building block of life. DNA is the blueprint that makes up a living thing. And sometimes an animal that went extinct million years ago, like a dinosaur, leaves behind blueprints for us to find. We just had an award. 100 million years ago, there were mosquitoes, just like today. And just like today, they feed on the blood of animals, even dinosaurs. Sometimes, after biting a dinosaur, the mosquito will land on a branch of a tree and get stuck in the sap. After a long time, the tree sap will get hard and become fossilized, just like a dinosaur bone, preserving the mosquito inside. This fossilized tree sap, which we call amber, waited millions of years with the mosquito inside until Jurassic Park scientists came along. Using sophisticated techniques, they extracted the preserved blood from the mosquito, and bingo, dino DNA. A full DNA strand contains 3 billion genetic codes. It's that long, and since it's so old, it's full of holes. That's where geneticists take over. Since most animal DNA is 90% identical, we use the complete DNA of a frog to fill in the holes and complete the code. Phew, now we can make a baby dinosaur.
Did you know DNA has a half-life of 521 years, and dino DNA that is more than 1 million years old would be hard to find with enough bases to create a baby dinosaur?
Did you know that DNA bonds will be destroyed after 6.8 million years?
Did you know amber is fossilized tree resin that is 25 to 50 million years old? Some of the oldest amber that has been found is 130 million years old. Amber has also allowed scientists to identify 1,000 different extinct species of insects and comes in 300 different shades, such as honey, cognac, citrine, cherry, green, and butterscotch. Did you know
Jurassic Park is impossible!
And here are the scientists to tell you why!
First off, there are a few problems in the extraction of DNA from mosquitoes and amber and then use of that DNA to create a whole dinosaur. DNA in amber would have already degraded a long time ago, leaving no trace to allow us to use DNA as a wholesome way to clone a dinosaur. DNA degrades after 5 million years, and the dinosaurs were at least 55 to 65 million years old. Some even dated back to 229 million years ago. Even if we were able to find a mosquito trapped in amber with DNA that hasn't yet been degraded, we would probably have gotten DNA that had been contaminated by outside sources. It has been known that outside sources such as plant DNAs can be mixed in with dinosaur DNA, leaving us unable to separate it.
Next, Jurassic Park stated that they used frog DNA to replace parts of the damaged DNA, and this is called a DNA buffer method. However, this process is extremely hard and normally works on recently extinct animals, such as a mammoth, which became extinct 10,000 years ago. Even if scientists were miraculously able to find dinosaur DNA, it would basically be impossible for scientists to fill in the empty gaps of DNA. Too many gaps would cause the animal to be more like, in this case, the frog instead of the dinosaur that we are trying to clone. The DNA we would have to get for the buffer method would have to be a close relative to the dinosaur, meaning that it had to be a species from 5 million years ago and has not yet evolved since then. This basically means that the buffer method would not work. However, if we had to use an animal as a buffer for dinosaur DNA, it would most likely be through the DNA of a dinosaur's closest modern-day relatives, the birds.
DNA is the building block of life. DNA is the blueprint that makes up a living thing. And sometimes an animal that went extinct millions of years ago, like a dinosaur, leaves behind blueprints for us to find. These blueprints can be found in salt deposits, such as a recent case where a 419 million year old DNA was found intact in ancient salt deposits. Scientists believe that the sample had been contaminated, but this was not so. A research team had found 6 segments of identical DNA that had not been seen by scientists before. There are missing parts in dinosaur DNA, so we must use the DNA buffer method. This means we take the DNA of a current species and use it to fill up the missing gaps. Since birds are the closest relatives to a dinosaur, we use the complete DNA of a bird to fill in the holes and complete the code. Phew, now we can make a baby dinosaur.